talking about not microbial genetics, but talking about genetics. You probably didn't call it genetics. Maybe you did. I will tell you, you'll probably be delighted to know there will be no Punnett squares in microbiology, which I really like Punnett squares. My daughter, when she was in biology, when she was a freshman, she was like, this is the word. I'm like, I love it. It's like a little puzzle. You get to put things like tic-tac-toe. We don't have to do that here. And spoiler alert, the reason we don't have to do that here is bacteria don't have two forms of their genes. They don't have multiple alleles for a particular trait. So we never, like what you see is what you get, and what you have is what you're going to pass on to your offspring. So no Punnett squares, right? So you talk about DNA replication, you talk about protein synthesis, and that's great. So knowing that, kind of like chapter 5, we're going to try to build on these concepts, particularly how they, work in microbiological systems. And we're going to spend some time, even before we get into genetics, talking about just hereditary material, right? Because genetics is a study of genes, but it used to be that people didn't know what genes were. And so we're going to spend some time talking about some really cool microbiological experiments that answered this question of what is the hereditary material of a cell, right? Because it turns out that in the early 1900s, and even up into like almost the mid 1900s, if you were to open a textbook and go to the genetics or heredity chapter, they would say, we don't know what offspring pass on to their children in order to make them look the same, right? Those alleles that Gregor Mendel talked about way beyond, we don't know, but we think they're proteins. Because there are so many different amino acids, and proteins have such variety, like only a protein could account for all of the variety of every cell on every organism in the whole planet, right? So that has to be protein. That's what we think it is, but we're not sure. And then there's this other thing, it could be DNA, but that seems very unlikely because DNA is just 4 nucleotides. And how could 4 nucleotides account for the vast variety of organisms on the planet, right? So in come the microbiologists who are like, challenge accepted. I would like to try to figure out what the genetic material of a cell is. So a guy by the name of Frederick Griffith, sad story, he died before people really understood how brilliant he was, which is kind of happens a lot for scientists, right? You get mocked for knowing things that other people don't know, and then all of a sudden you die and people are like, oh, it turns out he was really smart. Or like you're a painter, right? Like Van Gogh was like, a pauper. And, like you can't even talk about getting a Van Gogh for, less than millions and millions of dollars, right? So, Frederick Griffith in 1927 was doing experiments on a bacterium called Streptococcus pneumoniae. Guess what Streptococcus pneumoniae causes? Pneumonia, right? So, he noticed that there were 2 strains of this bacteria. So, strains of bacteria are kind of like breeds of dog, right? So they're the same bacterium, but they look a little different. So he noticed that one, when he grew it on agar, was small and rough in texture. And the other, when he grew it on agar, was large and smooth. So this is a colony of one strain, and this is a colony of the other, right? So grown at for the same amount of time, one is tiny and rough, and one is large and smooth, and so he called them rough and smooth. What he also noticed was that when he took these smooth bacteria, who by the way were smooth because they had a capsule, remember capsules, that's chapter 4, capsules are those things which help bacteria to survive, right? They prevent phagocytosis in the body, they prevent chemicals from getting into the cell, right? And when he took those encapsulated smooth bacteria and injected them into mice, not surprisingly, the outcome of that experiment was a dead mouse. This is why you're glad you're a person and not a mouse, right? Or that you're a person that wasn't born in like the 1700s, because guess what, if you were like an orphan, they would probably also, they wouldn't really do this to you, but they used to experiment on little kids, right? Because orphans had no parents to complain about it. So today we don't do that anymore. Mice, right? So mouse dies. Well, let's see what's going on inside the mouse. Not surprisingly to him, encapsulated bacteria were growing in the mouse, right? So then he takes the other non-encapsulated rough bacteria, injects them into the mouse, nothing happens, the mouse is fine. When he takes a sample from the mouse, he does find a few colonies of these rough bacteria have grown, right? So this is nice for two reasons. Number one, it establishes some experimental data that show us how well a capsule can protect bacteria, because they're just fine. No capsule, they don't survive in the body, right? There's very few of them. So this is not the wow of the experiment, right? This is just him setting up some controls, right? He's setting up some parameters, right? So the parameters are smooth bacteria kill mice, rough bacteria don't kill mice, right? And something in these bacteria tells them how to make a capsule, some genetic, right? Some hereditary material from those cells has been passed to the billions of baby cells there to teach these guys how to make a capsule. These lack that hereditary material, and that's why they're rough, right? So then he does another experiment, probably has an outcome that is not a surprise to you. And just so you don't think you're smarter than Frederick Griffith, it wasn't a surprise to him either. He killed encapsulated bacteria and injected them into a mouse, and because they were dead, do you expect the mouse to die? No, right? No surprise, mouse doesn't die. Also not a surprise that when you take a sample from the mouse, there are no bacteria there. So why does he do this, right? Well, what does heat do in controlling microbial growth? It kills them, but how does it kill them? What does it do to their enzymes? It denatures proteins. So he specifically is trying to kill the proteins of the cell, right? Because if he can kill the proteins, then those can't possibly be passed on to offspring. So he's trying to damage what is thought at that time to be the hereditary material of the cell. So he damages the protein, but because the cells are dead, no surprise to anyone, the mouse survives. So now he does something a little bit crazy. He mixes two non-lethal things in a combination. He makes himself a little Streptococcus pneumoniae capsule or cocktail containing live rough, which are non-lethal, and dead smooth, which are non-lethal. And he injects those into a mouse, and the mouse dies. And you're like, how? Like, how are two non-lethal things lethal? So the obvious next step is the same step that he's been doing. So he takes a sample from the mouse, and what he finds are live, smooth, capsulated bacteria. That is, and it wasn't a mistake, right? Of course, he went back and did this more than once. This is why we do replication in scientific studies. It's not a mistake. There are, every time he does this, he ends up with live, smooth bacteria. So he says, when he publishes these data in 1927, he says there is something transforming the rough bacteria, because they're the only ones that are alive, into smooth bacteria. Something from the smooth bacteria who are dead is being picked up by the rough bacteria, and it is transforming them into smooth bacteria. And he says, I think that it must be DNA, because the proteins are all denatured. So that wouldn't have done them any good to pick up denatured proteins, because they're not going to do anything for you. But DNA does not denature at very low temperatures, right? Or boiling temperatures, right? It takes a lot of heat energy to denature DNA. So he says, I think the hereditary material, the transforming principle, the thing that is going from the dead smooth bacteria into the live rough bacteria and transforming them is DNA. And he publishes those data, and people are like, whatever, like that doesn't make any sense at all. No, negative Freddy, no. And a guy by the name of Oswald Avery and his cohort of scientists pick up this idea, right? This transforming principle, because now it's 1944 and they have the capability of actually going in and trying to find this thing that's gone from one cell to the other, and they identify the transforming material as DNA. So they go, ka-ching, right? But it's sort of like the whole spontaneous generation, biogenesis, abiogenesis. You think you got the mic drop. You're like, it's DNA. problem solved, change the textbooks, and people were like, we're really not sure. It's gonna take a little more than your little experiment with bacteria to convince us the DNA is hereditary material. So, a few years later, Alfred Hershey and Martha Chase, this person's name was not Hershey Chase, it was Hershey and Chase, in 1952, right prior to the work of Watson and Crick. By the way, all of this work is what made Watson and Crick's discovery, and also the work of Rosalind Franklin, right, whose picture they stole. That, all of this, they would never have gotten the Nobel Prize if people thought in 1953 that protein was the hereditary material of the cell. Who cares what DNA looks like? We care because it's the hereditary material of the cell. Why do we know that? Because of Griffith in 27, because of Avery in 44, and because of Martha Chase and Alfred Hershey in 1952, who did a really cool experiment on, I won't say organism, I will say entity who is only protein and DNA. Right, because the bacterium, it's a cell, it's messy, it's got lots of stuff. It's got DNA, it's got proteins, but it's also got lipid inclusions and metachromatic granules and all these sorts of other things inside it, right? And so it's messy. So they think, here's what we're gonna do. We're gonna take a virus, because a virus is only those two things that we think are the possible hereditary material, right? It's either protein or it's a virus, or it's DNA. So they take these little viruses called bacteriophage, which are named bacteriophage because they were thought to eat bacteria. A bacteriophage structure is a protein shell surrounding a DNA molecule in its center. So they said all we gotta do is see which of those two things goes into the bacterial cell. Because whatever goes inside the cell is what's directing the cell to make viruses. So they designed two experiments. In the first experiment, they radioactively label the protein. So the whole shell is radioactive, right? So a bacteriophage kind of looks like this. They're kind of creepy little alien-like things, right? So the green is protein. The red is their DNA. So in this experiment, they've radioactively labeled this whole outside, right? The protein. So then they take those radioactive bacteria or viruses, and they allow them to infect bacteria, and then they look to see, is the radioactivity in the bacteria? Because if it's the hereditary material, the radioactivity will be inside the bacteria, and lo and behold, not only is the radioactivity not in the cells, it's in the fluid that never got inside the cell. So they say, okay, well apparently this tells us that the protein did not go into the bacterial cell to teach the bacteria how to make a virus. But just to hedge their bets, they do a second experiment where they radioactively label the DNA, right? Because you don't want an absence of a result. You want the result, right? So they do a second experiment. They radioactively label the DNA, right? So this red part is radioactive. They allow these viruses to infect bacteria. And then they look to see what got into the bacterial cells, and not only was there no radioactivity in the liquid, but all the radioactivity was inside the bacterial cells. So they published these data, and in 1969 they won the Nobel Prize because their data laid to rest that question, what is the hereditary material of the cell? The hereditary material of the cell is DNA, nucleic acid. Incidentally, if you go to the Smithsonian Museum, I don't know which one it is, I didn't realize until I went to Washington, D.C. a couple of years ago that there's more than one Smithsonian, there's like 87,000 of them. But in one of them, you can actually find this blender that these guys did this work on, right? Because what they did was they allowed the viruses to infect the cells, and then they put them in a blender so that it would disrupt them. And whatever was not inside the bacteria would get knocked away from them. So you can find these blenders, right? It's a regular old, like what you would make your smoothie in blender, and that's what they use. So now we know, because of microbiology, that the hereditary material of the cell is DNA, which is cool, right? Which is why we take some time to talk about it here, and you probably didn't take much time to talk about it in general biology. In general biology, you're trying to wrap your head around what an A and a T and a C and a G are, so you don't have time to say, well, how do we know that that's the hereditary material of the cell? You just say, oh, Gregor Mendel, wonderful, wonderful, DNA, boom, and here we are, right? So DNA, boom, here we are. The bacterial chromosome is just like yours in that it is double-stranded DNA. It is semi-conservatively replicated, meaning that when that DNA molecule replicates, one of the old strand goes to one daughter molecule and the other strand goes to the other daughter molecule. It's anti-parallel, A's pair with T's, C's pair with G's. Everything looks the same. In fact, you can give human DNA to a bacterium and he can replicate it and he can make proteins out of it. There's nothing distinguishable, right? You're not that cool. Your DNA ultimately looks just like their DNA. Now, what is unique about bacterial chromosomes? Number one, they only have one. It's double-stranded, but they only have one. Which means they're always haploid, if we think in those genetics terms, right? There's no such thing as diploid because there aren't two copies of each gene, right? You have 46 chromosomes arranged in 23 pairs, right? Which each of those pairs has the pair together have two copies for each gene, which is why we can talk about people as being carriers for traits. Bacteria are not carriers. One copy of every gene. They either have the gene, make it into a protein, do the thing, or they don't have the gene, don't make it into a protein, don't do the thing. So it makes it a lot more straightforward to work with them, right? So they have this chromosome, it's always haploid, it's also circular. Yours are linear. So bacterial chromosomes are circular and they are replicated at one single origin of replication. It starts here and heads in both directions, just like yours. Remember, you have to learn about all the fun stuff, the Okazaki fragments, and the leading strands and lagging strands. Don't worry, we don't have to do that again here. Super cool, but we're not going to study it here, right? The same thing, zip around, and at the end of that process, 2 molecules of DNA. And that's pretty much all we're going to say about DNA replication. You can have one little bitty thing, but you've already studied DNA replication, so I don't want to rehash that, spend the whole day talking about that again, right? So all we're going to say about the chromosome is, it's double-stranded, it's semi-conservative replication, A's and T's and C's and G's. What's new for you probably is that it's haploid and that it's circular. The other thing that is unique to prokaryotes is that in addition to a chromosome, right, because maybe you're feeling sorry for them. You're like, oh, that's so sad. You only get one shot at every gene, right? And it turns out, right, they don't have as many genes as you do. You have about 3 billion nucleotides. E. coli has like 3 million. You have 30,000 genes. He has like 3,200. So you're like, oh, that's really sad. But don't be sad for them because they have extra chromosomal DNA called plasmids. Plasmids are much, much smaller than chromosomes. They are also circular and they are self-replicating. They have their own origin of replication. So they have a little beacon where DNA polymerase goes, ooh, that looks like fun, and it comes over and it replicates the plasmid. And we're going to be talking a lot about plasmids. This plasmid here kind of has all the pieces of plasmids, right? So not every plasmid looks like this. These are some of the things that you could find on a plasmid. First of all, you will always find an origin of replication on a plasmid. That is the place DNA polymerase will come in and replicate the plasmid, right? You will have 3 major kinds of plasmids, the first of which is called an F factor. And an F factor stands for fertility factor. It's called a fertility factor, and we're going to talk a lot about this one in this chapter, because it allows for something called conjugation between bacteria. So conjugation, not like and or but in a sentence, but like a conjugal visit, right? Conjugal visits is where people have sex. Bacteria don't have sex, but when people saw this process under the microscope, they were like, well, that bacteria has a thingy that's sticking to that bacterium, and at the end of the process, that one has new DNA. So they're like, well, that kind of looks like sex, so let's call it conjugation. And when they figured out what was causing that, they're like, well, let's stick with that little idea, and we'll call it a fertility factor that's causing conjugation, right? So we're going to talk a lot about conjugation. But conjugative plasmids have this right here and this right here. So they have the ability to make a pilus, which we talked about in chapter 4. Remember, a pilus is that skinny tunnel through which DNA can be passed from one cell to another. So they have the ability to make a pilus and the ability to transfer this plasmid through the pilus to a recipient cell. So that's one kind of plasmid, is an F factor. And that's the one we're going to talk about the most in this chapter. A second kind of plasmid is something called a dissimilation plasmid. So a dissimilation plasmid is one that we're going to see again in our diversity unit, because dissimilation plasmids usually have on them this kind of thing over here. Resistance to something that is toxic or poisonous, and a lot of times the ability to catabolize that toxin, like eat it, right? Like instead of a cheeseburger, mercury. Or instead of a cheeseburger, TNT. Or instead of a cheeseburger, arsenic. Or plastic. Do you know there are how do you say that? Phthalate, I think. It's a plastic component in plastic. There are bacteria that eat that. And they can share that ability with other bacteria by sharing these plasmids with them. So dissimilation plasmids usually involve genes that will catabolize like typically non-food things into food. And then we have R factors. So it's a bummer, right? Because you think, well, we have an F factor and R factor. Why can we have a D factor? That's because someone else named it and they didn't want to be, they didn't want to be like everybody else, and they had to make things difficult for the rest of the world. But R factors are resistance factors. R factors are plasmids that carry genes for antibiotic resistance, which you can see here as tetracycline resistance, chloramphenicol resistance, streptomycin resistance, sulfonamide resistance. So those are different drugs and antibiotics that are used. And so if a bacterium has that kind of a plasmid, they are now not inhibited by that antibiotic. And so they will grow in the presence, and we would say, oh, now you have an antibiotic resistant infection. So these are not all found on single plasmids. What is found on all of them is an origin of replication. These two are on these F factors, these conjugated plasmids. These four are on R factors or resistance plasmids. And this would be an example of something that's found on a dissimilation plasmid. So they're just all of them drawn onto one plasmid so you can see it. All right, so they have chromosomes, they also have plasmids, but thankfully the way they use their DNA is just like yours, right? The flow of genetic information goes from DNA to messenger RNA to protein, right? We call that gene expression. Typically in general biology, you call that protein synthesis. But notice protein synthesis and gene expression, all you're doing is you're defining it from one angle or the other. If I say something synthesizes proteins, what I'm saying is a gene is transcribed into messenger RNA, and that messenger RNA is translated into a protein, and I have synthesized a protein. I could just as easily say I have expressed a gene, because the expression of a gene is protein. Just like the expression of a painter is a painting, the gene gets expressed as a protein. The gene is transcribed and translated into a protein. So in general biology, they usually call this protein synthesis, but when you get to other classes, a lot of times they call it gene expression, because it turns out that not every gene is translated into a protein, right? Some RNA just stays RNA, like ribosomal RNA, so they want to be a little bit more precise, right, with the language. When you're in general biology, it's just easier to go photosynthesis, protein synthesis, got it. Like that means I'm making whatever that name is that's connected to it, right? So we are going to call it gene expression. Gene expression happens when a particular gene is transcribed and translated into proteins, which of course allows the cell to grow, to replicate, to do its thing, right? There's also the process of DNA replication, right? DNA replication precedes binary fission, because you have to replicate the chromosome so that both daughter cells will have a copy of the chromosome, right? So these are both things that are not novel to prokaryotes, right? Gene expression and DNA replication are not novel. That everybody does that. But this is novel to prokaryotes. The idea of genetic recombination, where a cell, a bacterial cell, can collect information from other cells and incorporated into its chromosome and become recombined. Right, now that happens in the lab, right? Because everybody goes, ooh, genetically modified organisms, and you freak out about that. But I mean, it's a natural phenomenon. When we talk about that conjugation, one cell sharing DNA with another cell, that is a naturally genetically modified organism. That happens by itself. And because that's uniquely prokaryotic, that's where we're going to spend a lot of our time in chapter 8, right? And we call this horizontal gene transfer, right? Because rather than it being, for lack of a better word, parent to offspring, it is just you and somebody else around you. So what you actually call replication is vertical gene transfer, right? Parent to offspring. So thinking about it on a vertical axis, right? So parent cell with a chromosome passing on that chromosome, right? And in the next generation, we have daughter cells, and we call that vertical gene transfer. Recombination, we're talking about this cell sharing with an unrelated cell, right? Giving information, here we'll make his, right? So this guy, him sharing a little piece, and now he is recombined. He has his original green DNA, but now he also has a piece of black DNA from that cell. So this recombination that we're going to be spending a lot of time talking about, not through family, but sharing within the community, right? Unrelated cells. So not your offspring, but cells that you're just in contact with. This is a natural phenomenon, but it's also one that is a lot of times used in the lab to create genetically modified organisms so that they can, make maybe like human growth hormone for you, or insulin for you, or other things like that, so that you can survive in, diabetes or different diseases, right? So we're going to talk a lot about recombination. We're not really going to talk about vertical gene transfer at all, except I would like you to remember, just because we're going to want to put a pin in it, the only thing that I would like you to make sure you remember from general biology is that in DNA replication, it doesn't happen willy-nilly. It happens at particular places where a primer has been set. So in vivo, which is a term we use to mean in the cell, there's a little enzyme called DNA primase, or DNA-dependent RNA polymerase. Same name, same thing, two different names for an enzyme. Descriptive of different aspects of its role, right? So if we say DNA primase, what we're describing is what it makes for us. It makes a little primer so that DNA polymerase can come in, because DNA polymerase, right, it needs to be led somewhere. It can't just show up and start adding nucleotides. It has to come in and find a little piece of DNA that it can hold on to, or a little piece of RNA that it can hold on to. And in the cell, that is provided by this thing that's either called DNA primase, or more particularly, more specifically, precisely called a DNA-dependent, because that's what it's using as a template, RNA polymerase, because that's what it's making. It actually makes a little piece of RNA, and that little piece of RNA is enough to allow DNA polymerase to come in and to attach and start making new nucleotides. And if we blow up that area, what it's bringing in for you is this very important OH group right here, right? Because if we separate two strands of DNA, all you have is the bases, right? You have you've got your sugar phosphate backbone, but this is an A, and this is a T, and this is a C, and this is a G, right? That's what happens when you open up the strands of DNA. When you do that, right, DNA polymerase looks at it and goes, oh, that looks like DNA, but I can't do anything about it because I have to hold on to an OH group, right? Because chemically, that OH group helps to catalyze this reaction, right? Where a new nucleotide is brought in, right? Notice it's a T triphosphate, and that was an A triphosphate, adenosine triphosphate, right? All of these guys are triphosphates. So when he comes in, this little primer of RNA, right, it would of course be U, A, G has an OH group right there, and DNA polymerase goes, ah, that's what I needed. I needed that little OH group so that I can come in and make that next step. So why are we making a big deal about this right now? Because this phenomenon can be exploited in vitro in the lab, and we can direct the synthesis of DNA molecules anywhere on that 3 million nucleotide chromosome that we want to. All we have to do is design a primer that has that OH group, and DNA polymerase doesn't care. Doesn't care if that's a piece that was made by DNA primase or a piece that you made in the lab. As long as that OH group is there, DNA polymerase is happy to start adding nucleotides to the bottom of it. So we're kind of talking about this just to put a pin in it, because in chapter 9, we're going to be talking about making pieces of DNA. So for right now, right, because the test is going to end with chapter 8, what do we want to remember? Just that DNA polymerase needs a primer. All right. One other cool thing, before we get into the really cool thing about prokaryotic gene expression, is that prokaryotes can simultaneously transcribe and translate their genes, which is super cool. You can't, because those two processes are separated by a nucleus, right? Because where's the DNA in a eukaryotic cell? It's in the nucleus. So where's messenger RNA made? In the nucleus, right? Because what is the template for messenger RNA to make messenger RNA? It's DNA, right? You open the DNA and you make a piece of messenger RNA. If this is completely foreign to you, you might want to go and look back through some of your genetic stuff, right? But DNA, the gene, is transcribed into messenger RNA. In your cell, that piece of finished messenger RNA leaves the nucleus, goes into the cytoplasm, where ribosomes attach to it and start translating the protein. But in a prokaryote, there's no nucleus. So as soon as transcription starts, the ribosomes can go, ooh, there's a little piece of RNA, let's grab onto that and start making a protein out of it. So they can do these two things simultaneously, right? So here it's a little bit difficult to see, right? Because we're talking about molecules of RNA, which are tiny. But this yellow strand, that's the chromosome. These red blobs are all ribosomes that have attached to various pieces of RNA as they come off of the genes, right? So this is kind of a picture of that, right? So here's the piece of DNA. You can't see the RNA because they're all over the top of it. But you can tell, right, this one is longer, right? And because it's the whole sequence is covered with ribosomes. And so all of those ribosomes, not only are they doing it simultaneously, but then other ones can climb on and they can be making lots and lots of copies. So why is that cool? Because it allows them to respond to changes faster, right? If they sense something in the environment like, ooh, that's a cool thing to eat, I'd like to eat that thing, they might have to make the enzymes to eat that thing. But they can make that happen very quickly because they don't have to wait for the whole transcript to be done and move out of a nucleus and then get translated. As soon as they start transcribing it, they can start translating it, and then they can start eating it, which means lag time is small. Now, the really cool thing about prokaryotes, right? So prokaryotes, I said, have about 3,000 genes. Just like you, they don't need to use all of their genes all the time, right? Like you're very good at segregating what you turn on in your DNA and what you shut off in your DNA, right? Which is why you have skin cells and nerve cells and muscle cells and all sorts of different cells. Because in each one of those cells, you choose what genes to turn on and to use and what genes to shut off. So they don't have as many genes as you because they don't have to make nerve cells and skin cells and muscle cells and all fat cells and all of that stuff, right? They just need to make bacterial cells. But even those bacterial cells, they don't necessarily need, right? Because a lot of those 3,000 genes are for eating. But, you know, if they never see hamburgers, they don't need the genes for eating hamburgers, right? Because they're this unnecessary. So why make the enzymes to eat hamburgers if you live in the gut of a vegetarian and that person, you're never going to see a hamburger because that person's never going to eat a hamburger, right? So bacteria, like people, have the ability to regulate gene expression, right? To turn on certain genes when they need the proteins that those genes are expressed into, and to shut off certain genes, right? And they have varying levels of regulation, beginning with none at all, where you just have a gene, you transcribe it into a piece of messenger messenger RNA, excuse me, you translate that into an enzyme, and that enzyme is functional, and it takes substrate circle and converts it to product triangle. An example of that would be like all of the enzymes involved in glycolysis and the Krebs cycle, because you always need those, right, in the electron transport chain. You always need those proteins because you always need to be making ATP, and because you're always using them, you're using them and you're abusing them and they're dying, and so they need to be replaced, right? So there are some enzymes which are not regulated at all. They are transcribed and translated and are functional, and we call those constitutive enzymes. Always transcribed, always translated, and functional when they are translated into proteins. Now, let's say that it's a protein that you want to control whether it's on or off, right? So you may want to have it at the ready. You don't want to have to wait for that protein, but you don't necessarily want it on. So what you could do then is, so that you have it, you could transcribe it and translate it, but you could have it turned off. But that means you have to have a protein with an on-off switch on it. And you didn't maybe realize it when you studied this in general biology, but when you study allosteric inhibition, what you're studying is the on-off switch of an enzyme. When an enzyme has an allosteric site, right, that's not so you can do fun experiments in biology class. It's so that the cell using that allosteric site can either turn on the enzyme or shut off the enzyme. And we call that post-translational control. Because you've already translated the protein, we say post-translational control. The protein is present, but it can be turned on or turned off based on whether or not you need it to do its job. That's not a very tight control, right? And if you remember, and you don't have to because I said don't worry about it, right? But we talked about how many ATP it takes to make a piece of RNA, how many ATP it takes to make an amino acid. It's expensive. So you don't want to transcribe and translate that protein and just have it sitting off unless you need it at a moment's notice. In which case, it's worth the energy to transcribe and translate it, because as soon as you need it, you can turn it on and it's there. You don't have to wait for transcription and translation, right? So what if you're like, I don't really need it that quickly. I just, need it kind of quickly. So I'm not going to go to the trouble and expense of translating it and then shutting it off. I will transcribe it, but then I won't translate it. So this usually happens, you take the RNA and they kind of tie it up in a little bow so that ribosomes can't get on it. And then you have genes which are very heavily controlled and are very infrequently turned on, and only turned on when there's a need for them. And those are regulated at the level of transcription. So we have no control, constitutive, post-translational control, translational control, and these little babies over here you don't even transcribe unless something in the environment changes. Some change happens and that causes you to turn on the transcription, and these are called operons. And they are unique to prokaryotes. Now, you have a way of doing this, you just don't do it like bacteria, right? So with operon, this kind of transcriptional control is particular to prokaryotes. So hey, guess what we're going to talk about, right? So lots of stuff happen in eukaryotic cells which have these same effects, but they're not the same systems. So what we're going to do is talk about this transcriptional control, this operon control, and the nice thing about that is that when we talk about the operon, we also get to talk about this kind of translational control because it plays a role in the operon. So that's where we're going to spend most of our time, right? So you want to recognize post-translational, translational, right? But where we're really going to pay attention is post-translational and transcriptional control. So here is a situation where a cell will find itself undergoing transcriptional control when you feed them different kinds of sugars. So here, right, hopefully you're getting used to this. You're like, I don't ever want to see one of these again, where I'm looking at the log of cells over time, right? But that means that you're tracking a bacterial culture over time, much like you're going to be doing on Thursday. You could be doing with a spectrophotometer. You could be throwing this little tube into the spectrophotometer at metered intervals and watching it grow over time. And if you watch it grow on glucose, you get a steeper curve than when it grows on lactose. Steeper curve means growing faster, means faster generation time, right? They do that because they like glucose better. I mean, they really don't like it better, but we can kind of think about it like that, right? You feed them glucose, yum, 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 grow, 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 everything's great. Feed them lactose, they're like, I guess so, right? So then you wonder, well, what happens when I give them glucose and lactose? And if you're not looking at the picture in front of you, right, like if somebody asked me, what do you think growth would look like if you gave them both glucose and lactose? I would say, oh, it probably looks just like that, like somewhere in the middle. Because they'll just, they're just bacteria. They're just going to indiscriminately grab whatever's around, right? I mean, they're just little dumb guys. Well, here's the crazy thing. Here's what happens. When you give them glucose and lactose, they're like a little, you ever eaten dinner with a little kid where you're in a contest of wills about them finishing their dinner? And they eat like all the mashed potatoes, right? Mashed potatoes, gone fast. And then you sit there and you watch them like push the Brussels sprouts around their plate for like 37 hours, and you're threatening that they have to eat it for breakfast, and they're never gonna eat another thing again unless they finish those Brussels sprouts. And they sit there, and then finally they start eating the Brussels sprouts, but it's much slower, right? And they need 13 glasses of milk to wash them down. Mine actually like Brussels sprouts. My kids both get one food they never have to eat. My son is tomatoes. How dumb is that? I'm like, tomatoes? I mean, ketchup, yes. Pizza, yes. Spaghetti, yes. Tomato, negative. My daughter's is non-fat Greek yogurt, which is also stupid. But, all the things on the planet, that's what they would shove around their plates. So anyway, what's really happening here, right? What's really happening here is these bacteria are making a choice. They don't want to eat the lactose. They want to eat the glucose. They eat the glucose, yum, 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 yum. Why? Because they can bring glucose, split it in half in glycolysis, send it into the Krebs cycle, completely oxidize it, send all the electrons into the electron transport chain, translocate those protons, move them through an ATPase, make new DNA, make new proteins, make through everything, and boom, grow. And they will do that until all of the glucose is gone, and then they will sit there. But it turns out, right, remember we said lag phase, which is the first phase in a bacterial growth curve, is not nothing happening. Even though it looks like nothing's happening, there's actually a ton happening. And what is happening is they are preparing to eat lactose. Because all of the genes that are translated into the proteins to eat lactose are controlled on an operon, because they don't often see lactose. So here's what happens. They control this. These are constitutive. No control. They're always transcribed and translated, which means you're always ready to bring glucose into the cell. Lactose catabolism genes are located on an operon, and they are very heavily controlled, and they will only be turned on when lactose is present. Because only when lactose is present do you need to transcribe and translate those genes. And they are controlled on something called an operon. So these are a series of genes, right? Gene I, P, O, Z, Y, and A. Z, Y, and A, LAC-Z, LAC-Y, LAC-A, are genes for lactose catabolism. I'm not going to ask you to memorize that, but in case you're like, what does that mean? That's what that means. This P stands for promoter. This O stands for operator. And the I, dumbly enough, stands for regulatory gene. I don't know why, maybe because it was an inhibitor, I don't really know, right? But that's an op. So an operon has a regulatory gene, a promoter, an operator, and whatever you're controlling. So in this instance, we're controlling lactose catabolism, right? So a promoter, you may remember from general biology, is where RNA polymerase binds so that it can make messenger RNA. Notice that right next to the promoter is this thing called an operator. So imagine if RNA polymerase landed here and got ready to transcribe, and there was something big blocking its path. Then it wouldn't be able to transcribe the genes. Well, it turns out, so we're going to see in a moment, this gene is transcribed and translated into a protein that sits its fat butt right there on the operator and blocks RNA polymerase. And that's how we control whether or not lactose can be catabolized. Because the only time it can be catabolized is if RNA polymerase is able to successfully transcribe these genes, because those genes, once they become proteins, will bring lactose into the cell, break it apart, and send it into glycolysis. So what does that process look like, right? This is an example of what's called an inducible operon, right? Induced, like if I said to you, my sister is going to be induced today, what do you think is going to happen at the end, maybe tomorrow or today? She's going to have a baby, right? Because they're inducing labor. So we're turning something on, right? When you induce labor, you're making labor happen. So we induce an operon, we're turning on the operon. If we're turning it on, that must mean that its natural or native state is off. So normally, an inducible operon is off because that little fatty is sitting right on the operator. So we have to turn the operon on, and to do that, we have to get rid of that little fatty that's blocking it, right? So it turns out that this guy right here, that alternately looks like an M or a U, is a post-translationally controlled enzyme. It has an on-off switch. In the on position, it sits on the operator and blocks transcription. If you flip it off, it comes off and it can't bind, and now the inducible operon is on. So before you look over there, look here, right? The problem with operons is that you can get really screwy about what's turning on and what's shutting off. So what you have to do is this, you have to think backwards. I have an inducible operon, which means I have to turn it on, right? It has to be induced. So what is its natural state? Its natural state is off. This is where it gets crazy, because the repressor is on. So what do I have to do to the repressor? I have to shut off the repressor to turn on the inducible operon. Lots of offs and ons, right? So you have to think through it systematically, right? You have to think, right? It is inducible, which means it is off, which means I have to turn it on. How do I turn it on? I have to shut the repressor off, right? And once I shut the repressor off, it will turn it on, and that will make it work, right? But you can think all of those through if only you can commit your brain to inducible means I have to turn the operon on. So logically, what does that mean about the operon? It's off. So what does that logically mean about the repressor? It's on. So what do I have to do? I have to shut the repressor off, which will allow the operon to turn on and will allow me to transcribe and translate the genes to make lactose. So here is this operon. These are the genes for catabolizing lactose. Here's the natural, normal, we sometimes hear that called native state. Native just means what's normally happening, right? Normally, because this is further up, it is being transcribed and translated, and when it is transcribed and translated, it's turned on, right? It's in the on position. So it will go whoop and go right here, and it will block RNA polymerase, and none of these genes will turn on and be transcribed and translated. Now, let me ask you a question. You don't have to shout out an answer, you can just think about it. What do you think is the best way of figuring out when you need those genes, Z, Y, and A, to catabolize lactose? When do you know you need to catabolize lactose? Think for a second. Could it be when there's lactose present? Does that seem like too simple an answer? When do I need to catabolize lactose? When I have lactose, right? When do I need to catabolize a hamburger? When there's a hamburger sitting on a plate in front of me, or Brussels sprouts, or whatever it is. So here's the beautiful thing about an operon, right? It's completely self-regulating. You know what turns on the operon? Lactose. Which is beautiful, right? Because when do you need to do this and it won't be a waste of your time? When you have lactose. So lactose comes into the cell, and this is what happens during lag time. Lactose comes in and it hits the allosteric site of the repressor. And it changes its shape, and now it can't sit there. And so RNA polymerase will transcribe and translate these genes into proteins, which are enzymes, which will eat lactose. So when do you know that you don't need these enzymes anymore? When you don't have any lactose left. And guess what's going to happen to the repressor when all the lactose is gone? It's going to turn back on and it's going to shut off. So it's this beautiful system, right? It's like your toilet. Right? Like your toilet, how does your toilet know when to stop filling the tank with water? When the float comes up and it says, I'm full. And then you flush the toilet and it falls down and goes, I'm not full anymore. Okay, I'm filling, I'm full, stop, right? And it automatically shuts itself off. You don't have to do anything with your toilet unless it's not working, right? So this is a completely, wonderfully, beautifully put together system where lactose, and don't worry about allolactose, right? We're not in a biochemistry class. Where lactose regulates the system. When do I need the enzymes to catabolize lactose? When I have lactose in front of me. And that's what I do. And when do I not need them anymore? When the lactose is all gone. I ate it all. And that's what happens here. So it's very common for bacteria, right? Of those 3,000 genes, maybe 150 of them are all operons controlling the catabolism of a single thing, like lactose, or sucrose, or arsenic, or something like that. And they will not turn on those genes unless that food is available. And that way they're not making 1,500 different sets of enzymes, they're only making the enzymes for the food that they have in front of them. So this is an example of an inducible operon, right? And the effector, the molecule which changes the dynamics of the electron, inhibits the repressor, and that turns on the operon, and so we call lactose there an inducer. Right? Like Pitocin is the inducer of labor, lactose is the inducer of the lac operon. Here is, I wonder what I forgot to check on here is a video animation, kind of, from your textbook. Terrible animations. If you know biology and how to make videos, you can make a killing because you can make things that don't look this stupid. But I want to, all right, so hopefully the volume's up enough. But I want you to see this, and if you'd like to see this again, it's at the end of the chapter 8 notes. Because the operon is a dynamic process, it's kind of hard to see it as a picture, so I want you to see it happen. Hold on.
That the genes are usually not transcribed. When lactose is available to the cell, the operon is induced or stimulated to produce the proteins of lactose catabolism. The E. coli chromosome contains a regulatory gene near the lac operon. This gene is constantly transcribed and translated to produce repressor protein. The repressor protein binds to the operator DNA at the lac operator, physically preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for lactose catabolism cannot be transcribed into mRNA. When lactose is present in the cell, some lactose molecules are converted to allolactose. Allolactose binds to and inactivates the repressor proteins, preventing them from binding to the operator. The structural genes are transcribed, resulting in the production of the enzymes needed to catabolize lactose. Since allolactose induces or stimulates transcription of the lac operon, it is called an inducer. Notice that the inducer acts by turning off the repressor, not by actively stimulating the genes to be transcribed. When lactose is not available, the repressor proteins are active. The structural genes of the lac operon are not transcribed. This control prevents the cell from producing the proteins for lactose catabolism when lactose is not available.
All right, so just to kind of see that, albeit a little bit elementary, right? So notice, right, it's the presence of lactose. No more lactose, so the operon is turned back off. So if they're inducible operons who need to be turned on, what do you think there also are? Uninducible, yes, kind of. They're not really, but that need to be shut off, right? We call them repressible. So a repressible operon, the repressor does the same thing, right? The repressor is going to bind the operator. Here's the difference. Remember before in the inducible operon, the repressor was made with its switch turned to on. This one is made with its switch turned to off. So the repressor is normally inactive, which means it can't bind, which means it can't block the transcription. And so those genes are transcribed and translated. So a molecule will come in and bind to the repressor and turn it on, which will repress the operon. So repressible operons are on, so they need to be turned off, which means I need to turn on the repressor so that it will shut off the operon, right? So it's the same sort of, logical pathway. If it's repressible, it must be on, which means it has to be shut off, which means I have to turn on the repressor. So if you can just remember what repressible and inducible mean, you're there. But practice doing that little mental, logical step for yourself, right? So here's what a repressible operon looks like. So a repressible operon in its native state is transcribing an inactive repressor protein, which cannot block the operator. So the structural genes are transcribed and translated. And they're transcribed and translated, right, until some molecule binds to and activates the repressor, which blocks RNA polymerase and stops the production of those genes into enzymes or proteins or whatever it is that you're making. So this one's kind of cool, right? This is the trip operon. So that was the lac operon for lactose catabolism. This is the trip operon for tryptophan production or synthesis. So if we said lactose catabolism, we could say tryptophan anabolism, building tryptophan rather than breaking down lactose. So this set of genes are transcribed and translated into the enzymes to make tryptophan, which is an amino acid, right? So amino acids, right, when amino acids are made, they get connected to transfer RNAs and they're used to make proteins, right? So you would think that you would always want to be making tryptophan because you always need to make amino acids into proteins. So this is a constantly used thing, right? So you're like, yeah, that makes sense. I'm always going to make tryptophan because I'm always going to be needing to incorporate tryptophan into proteins, so I'm never going to have a stockpile of tryptophan in the cell. But what if it's Thanksgiving, right? This is everybody's excuse for not helping with the dishes, and I'm here to tell you it's bogus, so just help clean up. Because other people like to take naps on Thanksgiving too. Right, but turkey is high in tryptophan, right? So we go, oh, tryptophan makes me so tired. I eat so much turkey that I can't do anything. So let's say that you're E. coli living in the guts of somebody who just ate a bunch of turkey. So now you're sitting in the gut, right? They've got so much tryptophan they can't get all of it out of their intestinal tract. So some of it makes its way to the large intestine. And you're like, sweet man, because now I don't have to spend 3 ATP making every one of those suckers. I can just bring it in. And that's a much easier process, right? Just collect some tryptophan from the intestinal space or intestinal lumen. But when you do that, you don't need to make it anymore, right? So what's a great way of telling yourself to stop making it? To have tryptophan be the thing that activates the repressor. So when there is a surplus of tryptophan, either because the cell is not using the tryptophan they're making or because they're taking in tryptophan from the environment, if it's needed, it will go, right? It will quickly become part of charged tRNAs to make proteins. If it's not needed, it will hang around and it will bind the repressor, and now it turns on the repressor. And now the active repressor binds here, and no transcription or translation of these genes occurs, so you stop making tryptophan because you don't need to make tryptophan. When do you need to make tryptophan again? When all of your surplus is gone, right? When the turkey carcass is finally, after 17 weeks post-Thanksgiving, used up, right? So you will regulate this operon by the thing that you make, just like you regulated the inducible operon by the thing that you're eating. So that looks like this.
The TRP operon of E. coli includes a promoter, an operator, and 5 structural genes encoding proteins involved in the biosynthesis of tryptophan. The TRP operon is a repressible operon, meaning that the genes are usually transcribed. When excess tryptophan is available to the cell, the operon is repressed, ending the production of the proteins of tryptophan biosynthesis. The E. coli chromosome contains a regulatory gene near the TRP operon. This gene is constantly transcribed and translated to produce repressor proteins. However, these repressor proteins are inactive unless excess tryptophan is available. Once activated by tryptophan, the repressor proteins can bind to the TRP operator, preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for tryptophan biosynthesis cannot be transcribed into mRNA. When more tryptophan is available to the cell than the cell can use, some tryptophan molecules bind to and activate the repressor proteins. The repressor proteins bind to the operator, preventing the transcription of the structural genes that encode the proteins of tryptophan biosynthesis. This regulation prevents the cell from producing excess tryptophan when tryptophan is already available. When excess tryptophan is not available, the repressor proteins are inactive. The structural genes of the TRP operon are transcribed and translated, producing the proteins necessary for tryptophan biosynthesis.
Alrighty. Operons are my favorites. I think it's super cool. I mean, isn't that cool? I just love it all comes together things. Yes.
So the repressor proteins just hang out?
Mm-hmm.
Don't do it?
Yeah.
Why do you have to keep making, or is this just a symbolic?
No, I think it is like constantly made, but proteins, if they're not used, they get degraded, so they'll get recycled into this. So what'll happen, like if they're just sitting around not doing anything, is they'll get chopped back up into amino acids so they can be recycled. All right, another super cool thing that bacteria can do when they want to change what genes they're turning on is something called quorum sensing. So lactose metabolism, right? You induce that operon when you have lactose to eat. Tryptophan synthesis, you repress that operon when you don't need to make any tryptophan. But there's a phenomenon where bacteria can actually sense how many bacteria are in their environment, and that's called quorum sensing. Now, it sounds like a fancy biology word, right? A quorum sensing, but quorum just means a minimum number, right? You'll hear it in political settings a lot, right? It's the minimum number of like voting members required to actually take a vote, right? Because if only 2 people show up in the Senate out of 100, that's not a quorum, right? You can't just have 2 people voting on something that 100 people are supposed to vote on. So quorum just means a minimum number. So quorum sensing is when bacteria sense that a minimum number of friends has been reached, and then they induce behaviors. So they will behave one way when they are by themselves, and they will behave very differently when they have lots of their friends around. Sort of like bullies, right? Like bullies love to have like their little gang of dudes that are behind them when they're bullying, like Biff from Back to the Future, right? Like Biff loves an audience. Like he doesn't act like Biff unless he's got everybody behind him, right? And bacteria are kind of that same way. A lot of pathogenic behaviors of bacteria only happen when they have their friends with them. Some other things only happen when bacteria have a minimum number, right? So it's not a particular metabolite that's governing their behavior, it's that they sense their buddies are around, right? And they sense that chemically, because they can't see, they can't hear, right? So basically every cell creates chemicals and excretes them into the environment, and every other cell is sort of registering that, right? So that's that little triangle, that red triangle there, right? That's just, so when those chemicals are in very low concentration, you're all by yourself, so you better be chill. When you notice that there are tons of those chemicals around you, you know that all of your friends are with you, so you can act like a jerk, right? So for instance, Pseudomonas aeruginosa, who you work with, a non-virulent strain of Pseudomonas aeruginosa, by the way. Pseudomonas aeruginosa, when it initially infects a person, will be very quiet and just hang out. And it will just slowly produce babies, slowly making babies, but not making any waves. And then all of a sudden, when it's made its own little army, it registers that there are enough of them, and then it will turn on all kinds of virulence genes, which is really bad for the host, and that's you, because they have waited until there are enough of them that they can overpower your defenses, right? Which is why these can be really bad infections, right? And it's because they have changed the way they're behaving in response to there being more of them around, right? There are also cells who are capable of sporylating, right? Remember, it takes over 100 genes to be able to do that, so not everyone can. But you're not gonna sporulate all the time, right? Like if I took you, if you're Bacillus subtilis, and I put you into broth, and you're all by yourself, and there's tons of yummy stuff to eat, and tons of room, why would you sporulate? That would be stupid, right? You would grow. That's what you should do, right? You grow, and you grow, and you grow, and you grow, and you go for a while, and then you become competent, which we'll talk about later, and then it starts to get crowded. And now you're crowded, and you're swimming in pee-pee water, and there's not much to eat, and you think, you know, things are not looking that good, I think I'll sporulate. So they totally change their behavior. They shut down all of this stuff, this metabolic activity, and they switch to sporulation, right? Also, biofilms, right? You can't make a biofilm by yourself. So you hang out, and you reproduce, and you wait until there are enough of you, and then these cells will lose their flagella, land, and build a biofilm. But they won't do these things until it's crowded enough, right? So they're recognizing an increase in cell number, and that is causing them to change their behavior, which is cool, right? That's them responding to a change in the environment. And here, the change is not really anything external, it's just how many bacteria are present. And when there are more present, that will cause them to turn on these behaviors, and we call that quorum sensing. All right, one thing that we want to talk about with genetics, and it's sort of an odd, like, thrown in concept, and that is mutation. We talk about mutations for two reasons, and we're almost done for today, so just bear with me. Mutations are going to be our last topic for today. Most of this is general biology stuff, right? Who cares? Mutations are changes. They're not bad, they're not good, they're just a change, right? Now, normally those changes are bad because every organism is sort of fine-tuned to its environment, right? So a change is usually for the worse. Now, mutations can happen spontaneously. In about 1 in every 100,000 genes, no, 1 million genes, you get a spontaneous mutation, right? DNA polymerase is not perfect, it makes mistakes. But you have mechanisms, bacteria have mechanisms to fix those mistakes, right? That's one of the wonderful things about double-stranded DNA. If you make a mistake and you put the wrong nucleotide there, you can fix it because you know from the other nucleotide what should be there. Because if there's a T here, shouldn't be a G here. So you can cut out the G and replace it with an A, right? So you can repair those. But there are things called mutagens, which generate mutations. Now, I'm not going to ask you the numbers, right? I'm not going to have what's the natural rate of mutation. We don't care, right? But look at this. It goes from 1 in a million to as many as 1 in 1,000 per gene. So it really increases the rate of mutation, which overwhelms repair mechanisms and makes that mutation permanent, which could be a good thing, but it could also be a bad thing. And one of the things that we're gonna be doing in lab next week, nope, the following week, is exposing bacteria to ultraviolet light, which is a mutagen, and damages their DNA. And what we're looking for is the amount of time we have to expose them to ultraviolet light to where this happens. We overwhelm their repair mechanisms, and there are so many mutations that the cells die. But there are actually incidences specific to microbiology where we induce mutations on purpose. Because the mutation isn't necessarily bad, it's just a change, right? So when they discovered penicillin in the 1920s, everybody was super excited, right? In 1941, I think it was, they figured out how to actually treat infection with penicillin, and wow, that like changed the world, right? We're gonna talk about that in a later unit. But it turns out that that natural penicillin doesn't work against a lot of organisms, and there's a lot of resistance to it. So what they started doing was taking the organisms that make penicillin, right, because it's made by molds, and they started exposing them to mutagens like ultraviolet light. And what they have found, because of that mutant screening, is stronger, like a penicillin that's 1,000 times stronger than the penicillin that was discovered in the 1920s. And they found roughly 2,000 new antibiotic compounds just by taking, because antibiotics are produced by microorganisms. So they just kind of mutate them and see what happens. And thankfully they're little, so if we mutate them into like crazy little things, right, they're in a lab, in a petri dish, and we just kill them. So this allows us to find things that we can use therapeutically, right, to better success. The other reason we talk about mutations in microbiology is because, of course, mutations can lead to carcinogens, right? There are some mutagens that are also carcinogenic, and carcinogenic means cancer-causing, right? It's named for the cancer that was first discovered, right, with using this as carcinomas, right? So not all mutagens are carcinogens, and we don't want to worry about things that are not cancer-causing. We want to worry about the things that are cancer-causing, and we can use microorganisms as a step in that parade of identifying carcinogens. And the way that we do that is using something called the Ames test. So the Ames test, I think it's 1976 or 78, something like that, a guy named Bruce Ames came up with this protocol for identifying mutagens. And of course, when you're screening for mutagens, you don't want to rub them all over people, right? Because we don't want to do that to people. We don't want to do that to mice either, right? Because we like mice, they're cute, right? They're fluffy, they squeak, they're cute. So, you know, we just pick on bacteria because they're tiny and you can't hear them when they scream. So the first thing that we do is look for things that can mutate bacteria. And if things can mutate bacteria, then we say, okay, now maybe this is something we have to worry about. So then they put them into clinical trials with animals to see if they cause cancer in animals. But the first step to screen is in bacteria. So we expose bacteria to chemicals, and we look to see if those chemicals cause mutations, right? Because we can't cause cancer in bacteria, because bacteria is single cell. You either are alive and you're fine, or you're dead. So the way that they do this is kind of interesting, and the reason that it has to be this, because you look at the protocol, as soon as I can describe it to you, you're going to think, there has to be an easier way. But the reason it's as complicated as it is that you have to find a mutant that you can see. So you need that mutation to be a good one, right? Because if it's a bad one and they're not there, you're like, well, then it's not there. What does that mean? I don't know, right? So you actually want to induce a mutation that will allow bacteria to grow. So you have to start with bacteria that can't grow. So we start with what's called an auxotroph. An auxotroph, right? Trophic levels, autotrophs feed themselves, heterotrophs get fed. Auxotrophs need help to grow. Like auxiliary power is the help, right? It's not the main power. So auxotrophs, you have to feed them something. So in this case, Ames took salmonella and he mutated them so that they cannot make histidine. So the only way that they will survive is if you feed them histidine. So you take them, right? So you have these bacteria, and you know the only way that these guys will survive is if I feed them histidine. And then you grow them without histidine. So what do you expect to happen? No growth, right? I just said you have to have histidine for them to grow. You gotta feed them, and now you're not feeding them histidine. But even when you take those histidine-dependent auxotrophs and you put them on media lacking histidine because of the spontaneous rate of mutation, you will still get growth. Because in those two cells, a spontaneous mutation in that histidine synthesis gene fixed itself, and they were able to make their own histidine, which is pretty cool, right? So that's a lot of blah, 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 right? But what I want you to understand is they're dependent on histidine, right? And that's so that if there is a mutation, we can see it. It's visible as colonies, right? So if there are colonies there, there was a mutation there. So what's the baseline rate of mutation? In this example, 2. So you might have looked at this and you're like, okay, well that makes sense. Why did you grind a rat liver and stick that into the test tube? And the reason for that is, if I want to know what a chemical is gonna look like in your body, I have to see that chemical after the liver has processed it. Because that's what your liver does, right? Everything that comes into your body goes into your bloodstream, goes straight to your liver. Liver says, ooh, that's bad news, and then sends it back out, right? So we don't want to know what that chemical looks like to bacteria. We want to know what that chemical looks like to people. So we give it some rat liver extract, which means all the rat liver enzymes are there, taking the chemical and going and sending it out and exposing the bacteria to it. So it's part of the control. So in this one, rat liver, histidine-dependent salmonella, and the suspected mutagen. All together, rat liver enzymes are going to that suspected mutagen. Suspected mutagen is acting on those histidine-dependent bacteria. You put them onto this media lacking histidine, no one should grow, and yet billions of them grow. Because in each of those colonies, a mutation, right, and think about this, not just any mutation, somewhere in the 3 million nucleotides, a mutation that was common enough that it happened in just the right place in every single one of those colonies. So this allows us to screen, right? So this, we would say, yeah, that's a suspected mutagen. I would say that needs to go for further testing because it was able to mutate all of these cells. This is an actual Petri dish. On this upper side is the control. So there still are, right, some colonies because mutations are spontaneous. But down here, where the suspected mutagen is, we see lots and lots of those colonies, right? Not a few little white colonies, as we see up here, but right, dozens of little white colonies. And so we say, yes, this suspected mutagen is now a confirmed mutagen, but the question then is it a carcinogen? And we can't answer that with bacteria, but at least we can save the mice until they're actually necessary. So we talk about mutation, right, because mutation can be used beneficially to create antibiotics that are stronger, and we talk about mutations in general microbiology because we can use bacteria to identify mutagens, right, and keep people from getting cancer. And that is where we are going to stop for the day. And we do have a short lab, but I'll give you 20 minutes just in case you want to go grab some food or something really quick, and we'll start lab at